For a long time, when we talked about the biological clock and having children, the focus was almost entirely on women. It was a common belief that while a woman's fertility had a clear endpoint, men could father children well into their later years without much thought. However, new research is painting a much more detailed and nuanced picture. It turns out that a man's age does indeed matter, and it matters right down to the level of his DNA. The health of a man's sperm is a critical part of the equation for a healthy pregnancy and a healthy child. And this health can change significantly over the course of his life, which is something we are only now beginning to fully appreciate and understand. The reason this is so important is that sperm are not just simple cells, they are carriers of half of a child's genetic blueprint. This DNA is the instruction manual for building a human being, from the color of their eyes to the intricate wiring of their brain. Any small error or mutation in that instruction manual has the potential to cause problems. As scientists develop more powerful tools to look at DNA, they are discovering that these mutations accumulate in men's sperm as they get older. This means that the genetic information passed on by an older father can be quite different from the information he would have passed on in his younger years, with implications for the next generation. This topic is becoming more relevant every single day. Around the world, people are choosing to start families later in life. We've seen a significant shift in demographics, with the proportion of fathers over the age of 40 more than doubling in recent decades, from just over 4% in the 1970s to nearly 9% by 2015. This social trend makes the science of paternal aging particularly urgent. Understanding exactly what happens inside the testes as a man ages is no longer just an academic curiosity. It has become a vital piece of public health information that can help prospective parents make more informed decisions about their family planning journey and future. So what is actually happening to the DNA inside these sperm cells? The latest scientific research, published just this month in the esteemed journal Nature, has given us a much clearer view of how these mutations build up. Using a highly sensitive technique called nanosec, researchers at the Wellcome Institute and King's College London were able to analyze the DNA from individual sperm cells with incredible precision. They looked at samples from 57 healthy men, ranging in age from 24 to 75, giving them a wide spectrum to study. What they found provides concrete evidence that the risk of passing on potentially harmful mutations is not static. It increases steadily and significantly as a man gets older, creating a clear link between paternal age and genetic risk. The numbers from the study tell a compelling story. For men who are in their 30s, the researchers found that approximately 2% of their sperm carried what are known as potentially harmful disease-causing mutations. This serves as a baseline, showing that even in younger men, a small fraction of sperm will have these genetic alterations. However, this figure does not stay low. As men enter middle age, the rate begins to climb quite noticeably. The study pinpointed age 43 as a point where a more significant increase occurs, with the proportion of sperm containing these mutations jumping into the range of 3 to 5% of the total sperm population. This upward trend continues into a man's later years. The data revealed that by the time a man reaches the age of 70, the average proportion of his sperm carrying these potentially harmful mutations rises to about 4.5%. While that might still sound like a small percentage, it represents more than double the rate seen in men in their 30s. This doubling of risk is a crucial finding. It transforms the abstract idea of aging sperm into a quantifiable risk factor, showing that the genetic lottery of conception changes as fathers get older, with the odds of passing on a problematic gene variant slowly but surely increasing year after year. It is important to put these numbers into the context of the sheer volume of sperm produced. One of the most fascinating discoveries from this recent research is the phenomenon of selfish mutations. These are not just random copying errors. They're a specific type of mutation that gives the sperm stem cells a competitive advantage inside the testes. Think of it like a biological coup. A stem cell that acquires one of these selfish mutations begins to divide and replicate faster than its neighbors. This allows it to produce more and more copies of itself, all carrying the same mutation. Over time, this single rogue cell line can outcompete the healthier cells and begin to dominate sperm production within a particular area of the testis. This process, known as positive selection, means that these selfish mutations become far more common in the sperm population than they should be. The study identified 40 different genes where these selfish mutations tend to occur, and remarkably, 31 of them had never been linked to this process before. This is a significant expansion of our knowledge. Many of these genes are already known to be associated with specific health problems when mutated. This includes links to certain types of cancers and a range of rare congenital conditions such as Apert syndrome, which affects skull development, and Noonan and Costello syndromes, which cause a variety of developmental issues. The implications of this are quite profound. It means that the sperm of an older man may not just have more random mutations, but it might also have a much higher concentration of specific, particularly troublesome mutations. Because the selfish mutation gives the stem cell a survival advantage, it effectively creates a factory that churns out sperm carrying that specific genetic flaw. This explains why certain genetic disorders are more strongly linked to older fathers. It's not just a matter of chance. It's a systematic takeover by these aggressively replicating, mutated cells that steadily increases the presence of these harmful genes. This helps connect the dots between advanced paternal age and the observed increase in the risk for certain neurodevelopmental disorders. It is absolutely crucial to state that these findings should not cause alarm, but rather encourage awareness and further conversation. The presence of a mutation in a sperm cell does not guarantee that it will be passed on to a child, nor does it mean that a child who inherits it will definitely develop a disease. The journey from a single sperm cell to a healthy baby is incredibly complex. Many sperm with significant mutations may be less motile or unable to fertilize an egg in the first place. Others might lead to an embryo that is not viable and fails to develop, often before a pregnancy is even detected. The body has many natural quality control checkpoints along the way. As geneticist Matt Hurls, one of the study's senior authors, pointed out, the key takeaway is that fathers who conceive later in life may unknowingly have a higher risk of passing on a harmful mutation to their children. The overall risk for any individual is still low, but it is a factor that prospective parents may wish to consider as part of their broader family planning discussions. This research empowers people with more information, allowing for a more complete understanding of the biological factors at play. It shifts the conversation from one focused solely on maternal age to a more balanced view that acknowledges the role of both parents' ages in reproductive health. This field of study is far from complete, and many important questions remain to be answered by the scientific community. The next major step for researchers will be to track these specific mutations directly and determine their precise impact on the health of children. This involves conducting long-term studies that follow the offspring of older fathers to see which mutations are transmitted and what, if any, health effects they have. Scientists also want to understand why some men accumulate mutations faster than others, and whether lifestyle or environmental factors might play a role in accelerating or slowing down this process of genetic aging in sperm. Ultimately, this pioneering research opens up a new frontier in our understanding of human reproduction and heredity. By peering into the genetic code of sperm, we are beginning to unravel one of the most fundamental processes of life. How genetic information is passed and sometimes altered from one generation to the next. The more we learn about how and why these mutations occur, the better equipped we will be to advise families and perhaps one day even develop strategies to mitigate the risks. For now, this work serves as a powerful reminder that the story of our DNA is one of continuous change. with consequences that ripple across generations.